Welcome to Quicksand, the most important news in pharma and biotech this week. The turmoil at Canada's largest homegrown pharma company, generics maker Apotex, continues with the resignation of CEO Jeremy Desai, even as the company deals with the fallout from the murders of the company's founder. Apotex said that Desai had resigned and was being replaced by Vice Chairman and former CEO Jack Kay. The resignation of Desai comes 6 months after Teva filed a lawsuit against him and Apotex, claiming that for 2 years, Desai received copies of Teva trade secrets from his girlfriend, who before her 2016 departure was Teva's Chief of Regulatory Affairs for its American generics business. Separately, Toronto police announced that they were investigating the December 15th strangulation deaths of 75-year-old Apotex founder Barry Sherman and his 70-year-old wife as a targeted double murder. The Canadian billionaire was known to be litigious and was involved in a number of legal battles when Toronto police were called to the couple's 3-story home and found them strangled to death on the deck of their pool. It was reported that just days before they were murdered, Sherman had filed a lawsuit in an effort to quash a government investigation into whether lobbying rules had been broken when he held a political fundraiser for Prime Minister Justin Trudeau. He has also been involved in a decade-long legal battle with cousins who claim they are owed part of the Apotex fortune. Apotex has a history of issues with regulatory agencies. Following a November inspection, the UK's Medicines and Healthcare Products Regulatory Agency pulled the manufacturing certificate for an Apotex solid dose plant in Bangalore, India, saying investigators found a risk for cross-contamination. FDA granted fast-track designation to TAK-426, a purified, inactivated, alum-adjuvanted vaccine with whole Zika virus being developed by Takeda Pharmaceuticals. Last November, Takeda advanced the candidate into a 240-subject phase 1 study in the continental US and Puerto Rico. From the FDA's perspective, granting the fast-track label means the agency deems Zika a serious condition with an unmet medical need. There are no approved vaccines or drugs against the infection, which can cause microcephaly and other congenital brain abnormalities in infants born to infected mothers. For Takeda, the designation means more frequent interactions with the agency and eligibility for accelerated approval and a priority review. The Japanese pharma prioritized development of the candidate after the Biomedical Advanced Research and Development Authority committed $19.8 million to cover development costs through phase 1 and promised up to $312 million in assistance through a potential FDA approval. The Zika virus has significantly died down after an outbreak caught the world off guard in late 2015 and through 2016. Although the waning epidemic means fewer people are falling ill, the decrease in cases also makes it tougher to conduct late-phase clinical studies to prove vaccine efficacy. Sanofi cited that as one of the reasons behind its decision to drop out of an agreement to license a Zika vaccine candidate developed by the US Army. The FDA issued a draft questions and answer guidance intended to provide drug makers with more clarity about its Qualified Infectious Disease Product or QIDP designation. The QIDP designation was created by Congress to spur the development of drugs to treat infectious diseases in the Generating Antibiotic Incentives Now Act, which was passed as part of the Food and Drug Administration Safety and Innovation Act in 2012. The Act incentives include an additional 5 years of marketing exclusivity for drugs approved with QIDP designation and an automatic priority review for a product's first application for approval. Drugs granted QIDP designation are also eligible for fast-track designation, but sponsors must specifically request the designation from FDA. In the draft guidance, FDA clarifies that QIDP designation applies to a specific drug product from a specific sponsor for a specific use for which it is being studied, and does not cover an active substance in general or its uses beyond the qualified indication. FDA also noted that QIDP designation can only be applied to drugs and that most biologics and medical devices are ineligible for the designation or the incentives it provides. The European Medicines Agency released updated procedural advice on how it evaluates advanced therapy medicinal products, or ATMPs, such as cell and gene therapies. Alongside the updated advice document, EMA also released a new draft guideline on follow-up safety and efficacy requirements and risk management for ATMPs. According to EMA, the updated documents provide needed clarity to developers of ATMPs who are often small or medium-sized enterprises or academic spin-offs that may need additional assistance navigating regulatory processes. EMA's procedural advice document for ATMPs was initially developed following discussions among the agency's scientific committees in 2009 and describes the evaluation process and review timetable for ATMPs, as well as the roles and responsibilities of the various rapporteurs and committees involved in the assessments. By their nature, ATMPs are cutting-edge products whose safety and efficacy may not always be fully known at the time of approval. As such, EMA requires marketing authorization holders to follow a risk management plan and often requests that they complete additional post-authorization safety and efficacy studies. EMA says the revised guideline is meant to strengthen and clarify its requirements for post-authorization activities for ATMPs, building on its experience with authorized advanced therapies over the last decade. Specifically, EMA says the guideline provides new recommendations for detecting and mitigating risks and gives advice on post-authorization study design and methodology. Australia's Therapeutic Goods Administration has rejected the idea of using suffixes to differentiate biosimilars from reference products. Pharma trade group Medicines Australia backed a suffix-based model during the recent TGA consultation, but the agency concluded it would create complexity and potential confusion. Suffix identifiers were the backbone of one of the 4 options proposed by TGA in a consultation on the naming of biosimilars. The other options were to adopt a barcode system similar to that used in the European Union, encourage the inclusion of trade names in adverse event reports, or stick with the status quo. TGA asked for feedback on each of the options. Having analyzed the responses, TGA has established its position. TGA is sticking with the status quo in terms of naming, but is bolstering its oversight of biosimilars by implementing 1, possibly 2 of the other options. For example, TGA will make it mandatory to include the trade name of a product in adverse event reports, which will enhance TGA's ability to differentiate between safety issues that are specific to a product and those that apply to an active ingredient in general. The agency is making this the centerpiece of its near-term biosimilar strategy. TGA is yet to rule out adopting EU cell barcodes at a later date, though, and its response to the feedback notes its initial position does not preclude it doing so. Thank you for listening to Quicksand, episode 16, February 5th, 2018.